This talk is about embryology of the nervous system. This talk will be conducted using a question and answer format. The first question is: the hypothalamic sulcus separates the hypothalamus from the thalamus. A, true; B, false. This is a sagittal cut of the brain. We are going to focus on the area enclosed. Notice the hypothalamic sulcus. The hypothalamic sulcus is a diencephalic landmark. Above it, you can see the thalamus, and below it, the hypothalamus. Below and towards the midbrain, you can see the subthalamus. Posteriorly and rostral to the thalamus, we have the epithalamus. The structure I am just indicating is the medullary stria of the thalamus. I will now introduce several sketches to highlight several points regarding the embryology of this region. This is an old drawing of a 5-week-old embryo, brain, and rostral spinal cord. You can see the diencephalon, thalamus, hypothalamus, hypothalamus sulcus, epithalamus, metathalamus, a term no longer used to refer to the medial and lateral geniculate. This drawing is from the same book. It corresponds to a 3-month-old fetus. You can see the diencephalon, thalamus, part optica hypothalami, now just called hypothalamus, pars mammillari hypothalami, that corresponds to the subthalamus, and the hypothalamic sulcus. It was initially believed that the sulcus limitans extended to the forebrain. Hence, that the alar plate or lamina and the basal plate or lamina extended to the forebrain. Thus, in this version, the basal lamina contributes to the formation of the diencephalic structures. In such theory, the hypothalamic sulcus was nothing more than the continuation of the sulcus limitans. Later, it was considered that the sulcus limitans only extended to the midbrain. This theory appears to prevail at the moment. According to this theory, the diencephalon is formed from the roof plate, not labeled in this figure, and the two alar laminas, but not from the floor or basal plates. Based on this theory, the roof plate forms the choroid plexus and the epiphysis. The alar plate forms the thalamus, the subthalamus, and the hypothalamus. Yet more recent studies have cast doubt on this theory and put forward another theory. This theory explains the development of the diencephalon based on the presence of prosomeres, here labeled P1, P2, and P3. Guided by several proteins, but primarily by sonic hedgehog secretion from the ventral region and the zona limitans intrathalamica. These regions are shown in red and demarcated by the dark red lines. This theory put forward that the thalamus has been primarily formed by the second prosomere, the epithalamus primarily by the first prosomere, the subthalamus by the third prosomere, and the hypothalamus is formed by more rostral diencephalic structures. Regardless of the theory, it is important to identify the diencephalic structures in an embryo. Here you can see the cut of a 49 to 52 day fetus at the level of the brain. In it, we can indicate the dorsal thalamus, the ventral thalamus or just subthalamus, the hypothalamus, roof plate, and the foreplate. I have now indicated to the hypothalamic sulcus. Notice that as we have shown at the beginning of this talk, in the adult, the hypothalamic sulcus separates the thalamus from the hypothalamus and subthalamus, whereas at this embryonic age, on this view, the hypothalamic sulcus is caudal to the ventral thalamus. So the answer to this question is true. Next question. Which of the following structures does not have a C configuration? A. Caudate nucleus. B. Lateral ventricle. C. Hippocampus and indusium griseum. D. Fornix. E. Thalamus. In this frame, you can see the representation of a 35-day-old central nervous system. At this stage, all CNS division will grow. This growth is especially significant in the telencephalon. Yet, telencephalic growth is disproportional. Some areas grow more than others. For example, the area of the lamina terminalis, which, as you recall, is the site of closure of the anterior neuropore, hardly grows, and neither does the central region of the telencephalic vesicle. In the telencephalon, an area of maximal growth is the inferior mesial region, as indicated by the just introduced magenta arrows. Such a growth causes approximation of the medial region of the telencephalic vesicle to the diencephalon by 40 days, as indicated by the arrows in this new figure. By 45 days, the inner region of the telencephalic vesicle fuses with the outer aspect of the upper diencephalic structure as represented in this frame. In addition, forced by the disproportional growth of the different parts of the telencephalon, rotation of fastest growing region occurred to accommodate the growth to the space confined by the cranium. This growth and adaptation is such that, as we previously alluded, the upper and the lower regions indicated here, but also the anterior and posterior region not shown in this figure, grow disproportionately to the center area of the telencephalic vesicle. The consequence of these events is the creation of the insula and the opercular region, as well as the elliptical anatomy of some internal structures. I have introduced this photograph to illustrate the effect of decreased growth rate of the central telencephalic region, the growth of the surrounding area, and the restraint created by the cranium. Notice the initial configuration of the brain, the formation of the insula, and its gradual covering by the operculum, until the insula is no longer observed by looking at the lateral surface of the brain, as you can see indicated here. In this frame, I have introduced a lateral view of the telencephalon at about 45 days. The arrows indicate the direction of maximal growth. The cross line with the circle signals the center of the lateral side of the cerebrum. The lack of growth of the central region in the center of the cerebrum and the tremendous growth of the lateral surface of the hemispheres, especially in the frontal and temporal regions, as indicated by the arrows, leads to the C-like configuration of the lateral ventricles and many other structures. I will use this figure to highlight these changes. The C configuration is very noticeable in the lateral ventricle. The cingulum. The cingulum is a bundle of white matter compromised by many tracts that exhibit a circle shape nearly encircling the corpus callosum and extending from the rostral subcallosal area anteriorly to the temporal lobe. Another structure with this shape is the caudate nucleus, the dentate gyrus and the indusium grisaceum, the hippocampus and the fornix, the stria terminalis which connects the septal and anterior nucleus of the thalamus with the amygdala, the stria medullaris that connects the septal region, anterior nucleus of the thalamus, and the lateral preoptic hypothalamic region with the habenula, and the corpus callosum, here shown. Take a few seconds to see the labeled regions of the corpus callosum. Notice the position of the rostrum in relation to the splenium. So the answer to this question is E. Next question. Which of the following statements is not true? A. Olfactory bulbs develop from the lateral telencephalic regions. B. The ganglionic eminence gives rise to the neostriatum. C. The zona incerta develops from the telencephalon. D. All structures whose name include thalamus are of diencephalic origin. The cortex, also known as the pallium, includes the archicortex, paleocortex, and neocortex, as well as the cerebral white matter. These structures develop from the lateral telencephalic region. The ganglionic eminence, a lump of cells in close relation to the diencephalon that protrudes into the telesil, gives rise to the neostriatum, which include the caudate, putamen. From this area also comes the external segment of the globus pallidus, and according to some authors, the pulvinar nucleus of the thalamus. The lateral aspect of the telesil produces the lateral ventricle. Between the ganglionic eminence and the rostral encephalic mantle, we get the foramen of Monroe, and from the medial telesil, the anterior most region of the third ventricle, as represented in this added figure of an embryonic ventricular system. In this figure, you can see a portion of the telosil indicated by the area anterior to the yellow line forming part of the third ventricle. This area is very small, as you can see in the new figure representing an adult ventricular system. Notice that only the area anterior to the yellow line corresponds to the telencephalic contribution of the third ventricle. From the region of the diencephalon that fuses with the telencephalon develops the internal segment of the globus pallidus, the thalamus, the hypothalamus, which you can see indicated here, and the subthalamus, the area between the thalamus and the midbrain. In the subthalamus, we find the subthalamic nucleus of Lewis, the fields of Forel, the zona incerta. Also, from the same region, we get the epithalamus, which includes the pineal gland and the habenula. So the answer to this question is C. Next question. Which of the following structures appearance is not the result of advancement of cerebrocortical projection fibers to the spinal cord? A. Zona incerta. B. Nucleus subthalamicus. C. External segment of the globus pallidus. D. Internal segment of the globus pallidus. This image corresponds to a 2-month fetus. In it, you can see lateral ventricle, choroid plexus, hypothalamic sulcus, epithalamus, thalamus, internal capsule, subthalamus, and hypothalamus. This is a cartoonish representation of the same age cerebrum. Here I have represented the growth of the diencephalon. I have now introduced and labeled the 4 divisions of the diencephalon, and now represented a pyramidal axon destined to make contact with lower motor neurons. Here I am representing an advancing axon through the centrum semiovale and through the portion of the telencephalon that has fused with the diencephalon. Finally, the axon reaches the diencephalon and goes through between the thalamus and the subthalamus, as you can see here. In this frame, I have represented 2 changes. One is the axon going between the subthalamus and the hypothalamus, and the second change is the cerebral mass increase. I am now representing a second axon following the same trajectory, and now a third one following the same trajectory again, and now a fourth one. Notice that this axon is going through the subthalamic mass that now separates in 2 parts, an internal and an external mass. I have now represented a 5th axon separating the internal mass in 2. I will now proceed to name these masses: the zona incerta, the subthalamic nucleus of Lewis, and the internal segment of the globus pallidus. So the answer to this question is C. Next question. The dash communicates the thalamus with the internal segment of the globus pallidus and travels between the zona incerta and the subthalamic nucleus of Lewis. A. Fasciculus lenticularis. B. Thalamic fascicle. C. Subthalamic fascicle. D. Ansa lenticularis. This is the same cartoon we used in the last question. I have now added 3 lines. 2 of the lines indicate connections with the thalamus and the subthalamic mass, and the 3rd line indicates the connection between the different areas of the subthalamus. Here I have represented the consequence of the passage of the upper motor neuron axons on the way to the lower motor neurons. The zona incerta, the subthalamic nucleus of Lewis, and the internal segment of the globus pallidus. In this frame, I have shown further separation of the newly formed structures. The labels of pathways just created are color matched with the tracks. So we have the fasciculus lenticularis, the ansa lenticularis, the thalamic fascicle, which is the name given to the bundle of fibers destined for the internal segment of the globus pallidus as they exit the thalamus and before the bundle splits to form the fascicle lenticularis and the ansa lenticularis. The connection between the subthalamic nucleus and the internal segment of the globus pallidus is called the subthalamic fascicle. So the answer to this question is A. Next question. Neocortical neurons with long axons, projection neurons, are generated in an outside-in fashion. A true, B false. Once a neuron is generated, it approaches the spatial glia cells with a radial fiber that reaches the pia. The neuron moves towards the radial fiber, attaches to the radial fiber of a glia cells using an adhesive substance called C-aderine, and starts climbing till the cell is attached to the rod, as you can see in this figure. Then the neuron starts going towards the pia. In the next frame, I will show you photographic evidence of this process. Gino et al. In 2018 published a very nice photographic evidence of the importance of C-aderine. The control neuron was grown in a culture with no C-aderine added, and the photographs were taken as sequential intervals, as represented here. Here you can see the neuron, the line drawn from the lower region of the neuron at time 0. Let us appreciate the little climbing it does throughout the photograph. Now, similar neuron was photographed after adding C-aderine to the medium. As you can see here, the climbing neuron moves with a rather fast speed up and up towards the pia. Going back to our cartoonish representation of the neuron keeps on climbing, but once it reaches the pia, a detachment substance comes into play. At this point, the neuron loses its climbing apparatus and detaches, coming to rest at a given position in the cerebral cortex. Then the next neuron comes along and attaches to the radial glia, then starts climbing up and up and up till it reaches the pia, then loses the attachment, detaches, and occupies a space just above the first neuron. Then the same process goes on for the next neuron, climbing up and then detaching. The same goes for the next neuron and the next one. In the neocortex, here I have labeled the layers from 2 to 6 and added number 1 for a layer mainly of fibers parallel to the cortical surface where we find the Cajal red cell cells, especially in fetus and neonates. These neurons appear to be close to the pia prior to all other structures reaching it. The origin of these cells is unknown. So, as you can see in this sequence of figures, the first neuron to reach the pia becomes the deeper placed cortical neuron once all radial migration has taken place. This is referred to as the cortex being generated in an inside-out fashion. So the answer to this question is B, false. So deeper or inside neurons are generated before the superficial or outer neurons. So inside first, outside second. Next question. In a lab setting, an 8-week monkey fetus is exposed to a substance known to produce destruction of existing telencephalic commissures and to prevent further telencephalic commissure development. Which of the following telencephalic commissures will show signs of destruction? A. Corpus callosum. B. Hippocampal commissures. C. Anterior commissure. D. Posterior commissure. E. Habenular commissure. This drawing illustrates the cerebral commissures in an embryonic brain, yet only those of telencephalic origin have been labeled. The first telencephalic commissure to appear is the anterior commissure. It does so by the end of the second month. Next one to appear is the hippocampal commissure, indicated here and here, as it goes from one fornix to the other. This commissure appears at about 9 weeks. The corpus callosum appears during the 10th week. This commissure appears in the dorsal region of the lamina terminalis that has thickened and is now called the lamina reunions of His. I will now list the diencephalic commissures: the habenular commissure, the posterior commissure, the superior collicular commissure, the inferior collicular commissure, and the optic chiasm. So the answer to this question is C. Thank you very much for your attention.